the word for everywhere now with me now. The topic is about gene function analysis in plants and fungi. And it is held by three parties. One from Indonesia, right? And then we have another one from Egypt and from Thailand. And we have three speakers from each university, but all of them have one of the common thing. Uh, all of them are the alumni from the Göttingen University from Germany. They did their PhD together, and then with a good friend and good research, they all went back to their country and then still keep on relationship. And now we have the chance to listen to the research that they are doing, right? But before we get into the topic, we have delegates from each country to welcome and then just give you a little bit of remark before we get into the science of things, right? The first one, I would like to welcome Dr. Tun Siriputikit. He is an assistant president for international affairs from Prince of Songkhla University. So we call Ajan in Thailand, like for people to be a teacher, we call Ajan. So Ajan Tun, are you ready to give a little bit of a welcome and greeting to us all? Can you unmute, please? We have to unmute Ajan Tun first.
Yes.
Right. Okay.
Thank you. Finally, I can say something.
Finally, we
Greetings
can
to
hear you.
everyone. Yes, greeting from Thailand. And I'm here on behalf of the Vice President of the Suratthani campus, Prince of Songkhla University. He do send his regards and his remarks that today he would like to be here himself, but he unfortunately cannot. Congratulations on the tripartite collaborations from three universities from Indonesia, from Egypt, and Thailand. I'm glad to see the continued collaboration between friendships of PhD student from Germany, and we do welcome the current and the future. Any more collaborations between the research or exchange students or any other programs toward the improvement of the programs, including the genetic implants and fungi. So congratulations again, and hopefully today will go over very well, and I hope all the participants will receive certain something for everyone. And thank you very much.
All right. Thank you. So that was the greeting from the PSU. So now I would like to move on to ask Professor Asad Abdelkader Debala. He is the Vice Dean for Postgraduate Studies and Research from Tanta University from Egypt. It's still in the morning time, about 10 o'clock in the Egypt now. So please, Professor. Professor, we can't really hear you. Can you increase the volume a bit? We still cannot hear. So sorry, Professor Asad, we are still looking forward to hear. What can we? So maybe we can try again, Professor Asad. Let me check technically. Maybe I need to ask Audi to help me with this. So I think we will get the signal from Professor Assad afterward. So maybe when he come back, maybe we will go to a delegate from Indonesia. Professor Assad, can you speak something? Yeah, we can see you, but we cannot hear.
I think the volume of the audio, Professor Assad, you can check. Okay, we have a problem.
Yeah, I think it's
Atif, maybe you can try to speak in Arabic or to Achan, maybe to help.
For me, I cannot hear him as well. I don't know why. You hear me, Weedy?
Yes, we can hear you, Atif.
Yeah, but I misconnected with him as well here. Maybe he were here already. I don't know what is the problem with. Maybe I can phone him now. Just a minute.
So I would like to ask if maybe now I can invite Dr. Bohari. We will get back to Professor Assad later. Please, Dr. Bohari Yusuf from the Mullah Waman University. He is the Vice President of the university from Indonesia. Can you please greet and just a bit of remark for everyone? Thank you.
هو مش فاتح لك
Thank
ولازم
you,
نسمح لك.
Dr. Sutida.
بس انت
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon for Thailand and Indonesia. Good morning to Cairo. The Honorable Professor Asad Abdel Kader Derbala from Tanta University, Honorable Professor Dr. Karun Nakason from Prince of Songkla University, Honorable Baniat Certi PhD from Prince of Songkla University, Honorable Atef Abo Ogiala PhD from Santa University, Dr. Weedy Sunayo from Lama University, Honorable all participants in this seminar. First of all, on behalf of Director of Lama University, I express my gratitude for the initiation of this tripartite collaboration, especially to the Prince of Songkla University Thailand and Tanta University Egypt for the willingness to cooperate with Muhammad University. We are very happy to work with many universities in the world. We hope the cooperation that has been carried out between UNMOL and PSU since 2013, I think, can continue to improve both and especially for hope we can start cooperation this year, even though in the COVID-19 pandemic, this started with on this opportunity. this opportunity, let me introduce Mula Warman University briefly to all of you, especially to friends in Cairo and also in South Thailand. We hope that getting to know Unmul better and willing to make collaboration. Mula Warman University is located in Samarinda City, which is on the largest island in Indonesia, namely Kalimantan or Borneo, including the province of East Kalimantan, which is located right on the equator with a lot of natural resources and biodiversity. Its position is right in the middle of the Indonesia, which is one of the reasons why East Kalimantan will become the new capital of the Republic of Indonesia. So in the next 10 years, after the nation's capital moves to East Kalimantan, Mula Warman University will become the nearest state university from the capital. You can imagine we will develop rapidly. In Samarinda, we have one large campus and 4 small campuses. Besides, we have 300-hectare botanical garden and 22,000 hectare education forest. The vision of Unmul is development of science and technology, especially those related to the tropical rainforest and its environment, to become a center of excellence for tropical studies. Therefore, many research studies are being developed. For example, studies on natural products, medicine, cosmetics, social forestry, sustainable energy, and other related tropical studies. To implement this vision, Unmul has 14 faculties and 96 departments. And right now we have about 32,000 students and 1,200 lecturers. And in developing research, Unmul has 5 research excellences, such as the first is research on food, which includes biodiversity, local food, and its utilization. Second, the research on environment and natural resources, such as protection, utilization, and issues related to the environment and natural resources. Third, research is related to culture and information, such life harmonization, equality, and local wisdom. Fourth is energy related to protection and responsible utilization of non-renewable and renewable energy. And the last is research on health and medicine related to tropical diseases, tropical medicine, and use of biodiversity as a cure. Those 5 kinds of research, we hope that can grow together to reach the main focus is sustainable integrated tropical ecosystem-based development. However, we want the Prince of Songkla University and Tanta University to work collaboratively with many areas, such as staff mobility, credit transfer, a double degree, joint research and publication, student mobility, student exchange, degree and non-degree training, and others. Thank you to Songkla University for having collaborated for 7 years since 2013 with many activities with Mula Warman University. And my expectation to Santa University this year, we can start cooperation through the MUU, even though we make it out remotely, considering the COVID-19 pandemic that has not ended yet. And then we can immediately implement the collaborative activities in the following years. Thank you. I think that's I can say. Thank you and have a nice weekend. Wassalamualaikum warahmatullahi wabarakatuh.
Yes.
Salam.
Salam. Thank you very much, Dr. Buhari. I took your invitation very seriously. So hope after the COVID-19, we'll be physically visiting Indonesia. And also,
we are from Egypt. We will take this invitation and we will feel honored to make a collaboration.
yes. I would like to try if maybe Professor Asad can try to greet us.
I think the problem was him because every time he click in the mute button, I think some thing went wrong from the side of the host.
Oh.
The host has to give him the permission to go in, yeah?
Yes, but so at least we get to see you, and we know that you are here welcoming us, and that is great courage. Yes. So maybe the next one, with the time, I can move to the first speaker. So for a minute, that will be the first speaker is Assistant Professor Banyat Chim. He is the lecturer from the Faculty of Science and Industrial Technology from Prince of Songkhla University from Thailand. I have a very little understanding, but about his work, I think he will be able to tell us more. So without further ado, maybe Dr. Banyat, if you want to start. Please unmute your microphone. Dr. Banyat, can you please unmute your microphone?
Unmute. Okay. Because
Welcome.
I don't know Weedy or whom is the host. And then mute me.
We just to make sure that you know you are before you speak, so we just play a trick on you.
Okay, but everyone can see me, and I can say now in Thailand is 3:30 in the afternoon. I would like to thank first Weedy, my friend that invited me to be a speaker for today. Weedy is very nice friend from a PhD group, and when we finish, he also still in contact with me, even Atef. Okay, and then I think we should look forward to do the collaboration for the next future. Okay, I would like to share you my talk, and then share. Did you see? I have to.
Yep, we can see your presentation.
This is full. Okay, right?
Yep, it's good now.
Because this is my first time to use like a Zoom seminar for many participants, and have a moderator and more participants. and maybe something mistake. I'm sorry. But
No worries. You're great.
my talk is about basidiomycete enzymes causing industrial wood and its conventional identity. This is I would like to say that actually when I have done PhD work in Germany, I work with another kind of wood, like a softwood, because we cannot transfer like a hardwood, rubber wood, the rainforest wood. But I transfer the technique and the knowledge to Thailand, and then develop my work here. Okay. Also, I would like to introduce you that my university is it should be the first not should be, it is the first university in southern Thailand. And we have altogether 5 campuses, and the student nowadays more than 40,000. The first campus is Pattani campus, and Adai campus is the second. Surat Thani campus, Phuket campus, and the youngest one is Trang campus. That's why we have quite a number of the students. Academically, we can say we are a national research university and a comprehensive university. The ranking may be number 4 or plus, but I would like to show you the best one, number 4 in the nation in terms of publication. And the research strong points include natural rubber, biodiesel and energy, seafood and halal food, marine science, nanotechnology, peace study. This is Hat Yai campus, the second campus, but nowadays is the biggest one. And we just finished the graduate ceremony. That's why the Vice President cannot join with us. He has other work to do after this ceremony. And here, Surat Thani campus, my lovely workplace. I love it because everything happened here. Even my colleagues, my family, and not far from my parents. Surat Thani campus, actually, we have 2 faculties in the previous, and nowadays maybe they will have 2 more. And the faculty of liberal arts and management science, the faculty of science and industrial technology, that this my faculty, and have many departments. The red color here is my department, wood technology. And we are all together nowadays 6 lecturers, and one is the candidate. She is doing her PhD in Germany, and I'm a little bit proud because I'm the first one. And then why I can say I am the previous, the head of the department. The research field should be the conversion of wood to bioethanol, the extracellular enzymes on wood degradation by fungi, application of lignocellulosic enzymes on bioethanol production, wood modification, densification of wood, wood-based panel technology, biomass utilization, wood deterioration, energy from wood and other biomass, adhesion and adhesive, wood drying technology, equipment technology, or whatever. And we also have the wood science and industry technology research unit that have the aim to collaboration and knowledge exchange between wood science and industry technology researcher. The new knowledge transferred on wood science and industry technology to agricultural and industrial for sustainable international competition, international research publication, and to promote all research knowledge to public. Okay, this is my outline of my story that I will talk about my lovely place and transfer the knowledge and technique from Germany that I met very good friends like Weedy and Arabidopsis. Degradation of solid wood by fungi, the effect of fungi and fungal enzymes on wood, the contrasting ecological functions of wood extractives on protection against white rot, fungi, and induction of leachate activity. And I will close my talk with the application of wood for technical use in biofuel production. Everyone can also see here, this is my daughter. She's very small. I say small kids. I can say from here, small point to my big passion. Because I think everyone have a passion, but from some small point, that's why I love it to do this work. I love it to stay here. The left hand side is some kind of white rot fungi that is causing rubber wood. And also, if I count, since I have finished my PhD, more than 10 years of work on oil palm trunk and rubber wood. I applied for biofuel production, the possibility to use oil palm trunk. I can say OPT, and rubber wood sawdust as a raw material. The pretreatment and hydrolysis method for lignocellulosic materials, the methods for hydrolysis of polysaccharides. Actually, they have the stage of the development, the question of the rapid growing demand for energy. Maybe you can see nowadays the field is not much expensive in some country, but it's a bit expensive and it is limited resource on petroleum and gas, and the global warming by use of fossil fuel. The renewable energy sources is interesting because it is the first generation biofuel made use of food and fodder production, and the second generation biofuel that I call non-food material, such as lignocelluloses from trees and plants, give a potential to convert to be a biofuel and value-added product. Actually, we can make it like in just only three category, like a lignocellulose pretreatment to convert the recalcitrant lignocellulose structure to reactive cellulose intermediate. And we use the cellulose hydrolysis to cut it because we would like to use the sugar inside to make the biofuel. And fermentation. Maybe you can see from some text or some report, they have also more than my slide, but this is the main. If you come to Thailand, even in the southern part of Thailand, I'm very sad to to show this plant. In the middle, I'm happy with the rubber wood, but I'm sad with the oil palm wood because of they use the chemical to kill it and make it like a digest and then disappear, nothing used. The farmer also pay quite a lot per hectare. Two kinds of these plants is a fast-growing plants and then has a high potential sustainable wood production and application in wood products industry. I don't worry anymore about the rubber wood nowadays because they make a product. But for the oil palm, I think the researcher should do the research very hard to help the farmer to follow like the rubber wood. But one that I know in Malaysia that I have the last time a question, he is very good. I think it is the first one in the world make some product from oil palm wood. That is Mr. Chua, but I forgot his company, sorry. And the method this I use is the rapid test EN113 to use to measure the mass loss, how the fungi degrade wood, how much the fungi degrade wood. If you have this criteria, that means fungi grow very well. What is it? Water, temperature, oxygen, and wood. Wood is the food. So fungi grow very, very well. This I give a lecture for my student. All of every life come from this, because the human cannot produce food yourself. You have to eat plants. And just the water, carbon dioxide, and sunlight. You got glucose and oxygen. This is the photosynthesis system. This just show very fast that the oil palm plantation area is rise up very fast. And my students also have done the project with me very, very hard. Some lecturer nowadays, she also say, oh, very hard to work with the oil palm. But that student have done it. Make like this picture to cut into 3 parts, and then he measure. Because I would like to measure the physically and chemical content in this kind of wood. And this from rubber wood plantation, I think I'm not more worried about this. And the wood sample, left hand side is rubber wood, right hand side is OPT. Rubber wood, you can see if you cut it anatomically, is hardwood. It has a vascular abundance and then have other cells. But oil palm trunk is a difference. And the gradients of these vessel we call is also ordered in the different way. I also use these from my work since I study a master, and also transfer this technique to my PhD, and even I work here. I also use this. I think it is fine to do this, and it's work. And you can see here from the SEM that distributions of the vascular abundance from the bark to the center of the oil palm trunk is decreasing. That's why it is very hard to convert oil palm trunk to other products. It is not like rubber wood. It is the disadvantage point, but never mind. I didn't mind for that disadvantage. I try to use the fungi to degrade wood, and the diagram show you the Tripartite versicolor Ciputiana strain, the brown rot fungi that is basidiomycete. Basidiomycete, that means this kind of fungi secrete some enzymes and degrade the wood cell wall. That's why when these fungi grow on wood, the wood lost strength, lost chemical content, lost everything. Then I also try to do the experiment that found a brown rot fungus easily degrade wood than white rot fungus. The vesicular degrade late wood faster as early wood, something like this. Okay, go a little bit faster. The effect of fungi and fungal enzymes on a this is I transfer some kind of wood is in the group of softwood. This picture you also can see. Sorry. The first up is the control, and when I stain it with MBTH, you also can see a little bit brownish staining. And when you take a look closely within the wood cell, you will see what's happened with wood cell is stained with MBTH. That means this fungi do something with wood cell wall. And also this is my work that I told you from master to and up to now. And then easy for me. I don't know, maybe someone have the new technique up to them, but I use this red, you can see here. And then I stain it to change black and white, and then I count. And then I will know how big or how big of the area that the fungi degrade wood cell wall, like that. And something like the same from the plywood. And the lignin content of the wood block of control and the color, I compare here with white rod fungi, the color. And when I do the microscope and stain with MBTH again, you also can see here from the first 6 weeks, it gets red color. And lately, 12 weeks, 19 weeks, the wood cell wall is very, very thinner. And then would get like the would lost the strength, like that, something like that. That's why today I give a talk about basidiomyces, because it has like a tool to use the enzymes secrete into wood and degrade wood continuously like this. And I don't know, I would like to try more. That is functional or not. I try something like do by myself, inject like a enzyme into the wood and stain it. I found that it stained the cell wall here. Wood after 6 week incubation in the right hand side, but in the left hand, would inject with lactase. Lactase is the enzymes that fungi produce or fungi secrete. You can see also it can stain the MPTH to be red or brownish. And okay, that is visible in a reddish color. Modification of lignin results in phenol structure or whatever that is about the detailed, and the oxidative fungal enzymes can modify lignin within the wood cell wall. This is the white rod fungi that degrade wood because it degrades lignin. And lactase is my first interface or mechanism oxidase or whatever have also potential oxidized lignin within the wood cell wall. The contrasting, that's something different. Ecological function of the wood against white rod fungi and induction of lactase activity. I try also to put wood into the fungal liquid media and measure the lactase activity. About that, the black one here rise up very high, about 400 units per liter. What does it mean? That means wood have something to induce the lactase. The growth inhibition of fungi by the extractive. Actually, we use we can use water to extract some extractive from wood or acetone or whatever. And then the first one I see a very big of the inhibition zone here is a Arabidopsis cinerea, head cone in Thai, Protus austratus, head nang fa, and Dramtes versicolor here. And I would like to know that wood extractives protect wood. Is right or not? Untreated wood and extract with water acetone or whatever. You can also see here that when wood lost the extractive water extract wood, the mass loss rise very high. That what does it mean? That means wood lost the resistance to fungi. And just conventional technique to find out what kind of that chemical inside wood extractive. And I found that blah, 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 blah. It's too long. It has a short form. And I tried very small, very small amount. Fluoric acid, valulic acid. I would like to say very short like that. It's easy. And the behavior is the same. That means some kind of this extractive can induce the fungi to secrete enzymes to degrade wood. This is I never do before when I finished, because I finished physics from my bachelor, master in physics, but apply a bit in wood, and I have to do electrophoresis. Oh my God, very hard in the first, but I can do it and easily, you know. Just only make the gel run, and you make the mass spectrometry. And I found also what kind of the enzymes that the fungi produce. Is lactase 1, lactase 3, or whatever that we call. Okay, now I will go to the application of wood for technical use in biofuel production. This is my last of my talk. And actually, I also have done with another group, like the professor from China, from South Agricultural University, SCAO in Guangzhou. But they do also the another treatment, and then try to measure the durability to fungi or mold or whatever. But I never leave about the energy, because I think I love it. I have done for more than 4 years, and then yeah, maybe it stay in my brain, stick there. And I found that the conversion of cellulose to glucose, I only give you this. Whatever you use the glucose as fermentable to be ethanol or whatever. But when we use this cellulase and salinase and lactase, you can see it can convert the cellulose to glucose close to 100%. What does it mean? it can decrease the inhibitor, like it can decrease the inhibitor like extractive from wood, like that I can say. And then this is bioethanol production, improvements of OPT pretreatment I also have done with a decay fungi. And I found the cellulose in wood can be converted into fermentable sugar by cellulase. The pre-treatment of the wood is required because if you would like to use the cellulose like something is covered with lignin, you have to peel it out like you would like to eat banana. But maybe someone eat a banana with a bark, but I don't know. But for me, I never. I eat the meat inside, peel like that, and then you eat meat like that. Pre-treatment of wood white required, and the microwave pre-treatment is advisable by the lower energy and lower the acid. And in enzymatic cellulose conversion, the lactase helps to degrade compounds inhibitory to cellulases. And the treatment increase also this the physical property, like make something bigger, fivefold, and make it like maybe is the advantage for use the chemical to preserve it or something like this. I also found here treatment detectable increased absorption rate of the water. And again, come to the energy. Calorific value improvement of oil palm trunk and rubber wood pellet by decay fungi pre-treatment. I told you already, I never leave it. I come back to energy again, not a liquid like ethanol fermentation and to get the like a bioethanol, but these are solid calorific improvement found by chance, my student. Why they didn't use oil palm trunk to produce a wood pellet? Have many disadvantages. I think everyone here maybe know, but by chance it changed the chemical content inside, and then she found that the calorific value rise up. That is the very good result, and comparable to the rubber wood. But for the physically like oil palm trunk have a very hard fiber or whatever. There may be we cannot cut it easily, or we cannot use the saw the same saw with a rubber wood or whatever. But this is not my work. Other one should work. I work with this. Okay, I think it should be about 30 minutes, Dr. Sutida.
Yes, you are precisely very much on time. I'm really scared.
Okay.
Like to control like everything.
Because I put my watch here, but someone also can prepare the slide, and then they should know the countdown. I use the old-fashioned. I don't know, but I would like to say thank you for all audience or participants or students or people that's interested in our topic, genetic fungi. And I would like to thank to my supervisor, Professor Cruz, that once she have visit me in here at home in Thailand. And I would like also to thank to all of my colleagues. Thank you for your attention.
Thank you. Thank you very much. So I would like to thank you, Dr. Banjat, for sharing his research data and information on like all of the 4 interesting topics. And now I understand like how we are peeling bananas, just like that's how you pre-treat the cellulose before you get the sugar.
Yeah.
This is like very pictorial.
But someone told me that easy like a peel banana and eat.
That's a good attitude.
Think back, someone peel from the top, someone break in the middle, someone, I don't know, peel three pieces, someone peel 5 oaths.
So different way of peeling it.
Yeah.
Right. So
not easy.
That's good. Interesting. So that would be the talk from Ajahn Manyat. And if any of you have the question, you can feel free to type in the chat box. And then we will have the question and answering session afterward. Next, I think we are going to listen to the next talk. Our next presenter is Associate Professor Atif Abo Ogiala from Tanta University. So, and before he start his talk, I would like to introduce his background to all of you a little bit. Associate Professor Atif, he finished his bachelor and master in science, and then he finished his PhD in plant molecular biology from the Göttingen University from Germany. His major research interest is in horticulture, and the sort of like minor interest, but is the vegetable production under different system. So his talk will be slightly off from the science. I think he will focus a bit on the strategy, like the alternative option that we have. His topic will be how to find an alternative strategy for gene transfer to help plant against weather change. So let's hear more from Professor Atif, if you are ready to share with us. Professor Atif, you need to unmute your, and Ajahn Manjat, can you please stop sharing your file before Professor Atif can carry on?
Okay.
Yep. I think you need to click stop sharing in the Zoom, Ajahn Banjat. Yep. So now the floor is ready for Professor Atif after he unmute his computer. Professor Atif, can you hear us? Professor Atif? I hope he doesn't decided like, no, I'm leaving. Please wait a few minutes before he come back. Yes, I think we are going to see his presentation. He's sharing the screen with us now. Professor Atif, can you hear us?
Atif, do you have any problem?
I cannot see Professor Atif in the Zoom room.
Really? Yes, yes.
All
Please
right.
unmute, please unmute Atif. Aldi, tolong dibuka Aldi. Atif, don't click anything. Aldi will help you to unmute.
So Professor Atif, I think he's trying to unmute the system. We need
because this is the time like it means that yes, Atif, you can try to speak. Oh, still unmute.
Yeah.
خلاص.
Okay.
Right.
Okay, now because the problem was by host, not by me. He has to, the host give me the permission to unmute, yeah? Because
right. So we have you now, and then
please try to share your screen.
Yeah, he's sharing his presentation.
yeah, sure. You hear me?
Yes, we see your presentation now. But make it full screen. Excellent.
So I can, yes, I can start now, or?
Yeah, please.
Okay, hello everybody here. First of all, I would like to express about my deep thanks to Dr. Weedy Sunayo for this invitation and giving me the chance as a speaker in this web seminar. And it was also a good background I have collected with him in journey and with my other colleagues, Dr. Pat. And we still have a good relationship since 10 years ago till now. So I'm so happy to be here today. I will talk today about somehow an interesting topics to all of us. But before I start, I would like to introduce myself again. My name is Atif Abu Ugiala. I'm working as associate professor at Horticulture Department, Faculty of Agriculture, Tanta University, Egypt. And my talk today will be how to find an alternative strategy to gene transfer, helping planets against climatic change. First, I would like to introduce Egypt to all of you. Here, this red area you can see, this is Egypt in North Africa. Here, this is the white Mediterranean Sea, and this is the Red Sea. And this area with red color is Egypt. And this is a closer photo for Egypt area by Google Earth. Here is the green one is Nile River, which divided in Cairo city to two branches, making Delta Nile. This is Delta area contains many cities. Here exactly in the middle with white dot here is Tanta city, almost in the middle distance between Cairo and Alexandria. And here is again is the white Mediterranean Sea, and this is the Red Sea. Interestingly, Egypt has two of the 7 wonders in the world. First, pyramids in Cairo, and the second, lighthouse in Alexandria. So it's very interesting to come to Egypt for visiting, and you will enjoy visiting very nice places like this ancient civilization places, and this also tourism places like Sharm el-Sheikh, Alexandria, and Hurghada, and so on. So you are welcome to visit and enjoy Egypt. Coming again to the title of my topic today, how to find an alternative strategy to gene transfer, helping planets against climatic change. Actually, my goal from this presentation is to pay attention of all researchers to another side of improvement plant adaptation to abiotic stresses, which strongly correlate to climatic change. Gene transfer is the most modern method for researchers up to date for many purposes. And along the past 10 years, I collected huge experiences in this area. Everybody is here with me? Hello?
Yes, Atif.
Yeah, I can continue, okay?
Yes.
Okay. So again, my goal from the team is to be of all researchers on the side of which is strongly to climatic change. It's known, of course, that gene transfer is the most modern method for research update for many purposes. And along the past 10 years, I collected huge experiences in this area. And it is very well known that most studies in that area always focus on single genes that can be induced by specific abiotic or biotic stress. The study with interested genes takes a long way, starting by bioinformatic analysis using special programs like Gene Investigator, or even the internet tools for looking homologous genes in all organisms with high similarity to confirm the response of interested gene to defined condition before starting the laboratory experiment. Here, for example, show the result analysis by Gene Investigator program where it shows the regulation of the interested genes under different conditions, either in plants or other organisms. The right side here of this histogram shows the upregulations of the interested genes. The first column, this one, shows the different conditions that gene responds to it. This is to show you how the results of Gene Investigator analysis look like. After that, we look at the gene structure in gene bank data that recorded by the scientific institutions like Salk Institute. This to know the exact position of interested genes. Here, I have worked with 4 genes like says BC1 and SC5, and this is the structure of each genes containing the different regions, exon, intron, and all located between 2 untranslated region here. And here, this is the exact positions of the tDNA insertion within the structure. And each gene has 2 knockout mutants for the study. So these are photo for gene electrophoresis for PCR of the genes very much confirms the exact position of tDNA insertion within it. This also are photo of Western blot analysis that confirm the expression of generating overexpressor lines
I think Professor Atif,
I think he has problem with.
yeah, I think maybe the internet problem.
We have 259 participants. Great, very nice.
Maybe Professor will try to re-enter the meeting again, I think. We can wait a little bit.
Yes, Atif already there. Please continue your presentation.
Yes, I think Professor Atif is back into the room, and he's going to share his presentation, I think. Probably just now he take a short break. Yeah, so we can carry on with the presentation now. Professor Atif, you have to unmute your computer. We can see the presentation at the moment, but we cannot hear you.
Okay.
Yes.
Now.
Yeah, it's all good.
So my question is where my presentation was interrupted. Yes, where exactly? I don't know, but I can, bring my audience quickly again to my talk. Okay.
Yep.
Okay. So I would say that the most studies with gene transfer strategy, the most studies with the gene transfer strategies always focus on single genes, not a set number of genes. Single genes can be induced by specific abiotic or biotic stresses. The study with interested genes takes a long way, starting by bioinformatic analysis, using a special program like Gene Investigator, or even via internet tools for looking the homologous genes in all organisms with high similarity to confirm the response of interested gene to a defined condition before starting the laboratory experiment. Here, for example, the results of the Gene Investigator analysis, where in the where it shows the regulation of the interested gene under different conditions, either in plants or other organisms. The right side of this histogram shows the upregulation of the interested genes, and the left side shows downregulation of the interested genes. The first columns show the different conditions. This shows the first conditions is the gene response to it. After that, we look at the gene structure in gene bank data that recorded by scientific institutions like Salk Institute, this to know the exact position of interested genes. Hello? Everything went well, yeah?
Yeah, you can carry on. We can hear and can see the presentation well.
Okay, so this photo shows the structure of the genes under studies, like TIL gene, says gene, DC1 gene, SCL5 gene, and the structure of each gene, of course, has many regions, exon and introns, and all located between two untranslated regions. And here is two tDNA insertion mutants or sequence which generated knockout or of the every gene. So here is exact position of knockout mutants of each gene, and this. After that, we look at the photo of gel electrophoresis for PCR analysis of interested genes using specific primers to catch and confirm the exact position of tDNA insertion within the genes. Here is for 3 genes, 2 lines of each genes. This is for TIL genes, for example, and this is for the cis genes. And this is with left and right primer, and this is a specific primer for the tDNA LBA1. And this also photo for Western blot analysis that confirms the expression of generating overexpressor lines under control condition and salt condition. And you can see here that weak band under control condition, and after salt exposure it was clear bands, or shows clear bands here, reflecting the expression of the overexpressor lines used in that studies. From the next step, also we had to plant, cultivate for characterization of these genes under the study for phenotypic studies and physiological and biochemical studies. In parallel, transgenic lines of each single gene can be generated once as overexpressor or slice lines to confirm the role of each single gene in adaptation to the stress condition. Okay, coming closer to my message here today, which is that abiotic stress inducing a huge number of genes that work together to help plant adaptation to such stresses. And we must pay attention to that fact, not just to a single gene. And interestingly, I found in review literature some researchers who supported this idea and started their studies by subjecting a number of abiotic stresses at the same time and figured out different effects of the combination of plant growth and production compared to each single stress. This exactly reflects that set of genes can work in different directions compared to each single stress. For example, Kels and Ansel 2002 found that combination of salinity and heat stress in wheat were led to excess of damaging compared to individual stress. Girard et al. 2008 found that interactions between highlight and drought were resulted more damaging effects than individual ones. Ahmed et al. 2013 found higher negative effect on cultivated barley due to combination of salinity and drought more than the negative effect. Ahmed et al. Cultivated barley due to combination. In contrast, Perez Lopez et al. 2013 reported that interaction between high temperature and salinity increased the accumulation of osmoprotectants such glycine betaine and trehalose, which play a role in protecting plants against this stress condition. In publication 2019, I found promising results to the foliar application of generating molecules under stresses either individually or in combination for promoting pomegranate plants adaptation with set of environmental stresses happen together. This application titled with managing crop production of pomegranate wonderful via foliar application of ascorbic acid, proline, and glycine betaine under environmental stresses. My idea in that publication started from the end product of the genes, which inducing generating the material used in this investigation under different abiotic stress, where the material used ascorbic acid, proline, and glycine betaine. These material were the most highlighted molecules that many pathways of huge number of genes ended with it. In addition, most of biochemical studies under abiotic stresses using these molecules as an indicator for stress studies. Here, for example, this module explains the different pathways of genes ended with ascorbic acid. We can see and find many genes with red circle here, like BG1, who's responsible for generating D-glucose that converted to D-fructose, and BMI responsible for to convert fructose to D-mannose, and so on, till the set ended to ascorbic acid via collaboration set of genes, as in here. So many genes, BGI converted glucose to fructose, BMI converted fructose to mannose, BMM to mannose 1, again P, GME, galactose, and so on, from molecule to other molecule till the ended with ascorbic acid. So generating ascorbic acid molecules can be generated and promoted by several genes here. And of course, it was interesting to find the artificial ascorbic acid, which we can use it as foliar application to help plants from the end to overcome the stress conditions. Also, in proline product, located many genes with circle, with red circle here, like B5CR, B5C, B5CS1, B5CS2, B5CDH, B5C, and so on. Many genes locate the best ways and share together, work together to generating proline. And we have also proline as artificial material, and we can use it for foliar application under stress condition to help the plant adaptation to that stress. Glycine betaine as well. It's also located or at the end of the pathway of many genes like OPU, PA, PP, PC, PD, GBS, other gene, GBSR, A, P, OPU, C, T, and so on. So many genes also work together under stress condition for generating glycine betaine, like proline and like also ascorbic acid. So the idea comes from the end. Of course, the end product of the inducible genes is this glycine betaine or ascorbic acid or proline, and we use this idea to use this external foliar application under stress conditions to pass and short this expensive strategy for gene transfer, and of course cost as well. Here is the design of my experiment in that publication, which published in 2019, explains the concentration used from each material according to the recommendation from previous studies. For example, 10 millimolar of ascorbic acid were used, and of course 50 millimolar of proline were used, and 100 millimolar of glycine betaine were used, and the combination between them. These concentrations arranged randomly in 8 treatments in one plot with 3 replication of each. And the result was promising, and we can see from this table that the most promising results we have got from the examination of this material, ascorbic with proline with glycine, where it shows that yield of the pomegranate fruit and the fruit weight also were the best, and fruit retention. And also these 3 combinations also limited the negative effects of which can happen on the plants, like fruit cracking or sunburn fruit. And
so
these are references of my
Professor Atif, I think do you need to move the slide? Oh, is he? Oh, maybe I shouldn't interrupt him, but I want to see the the PowerPoint. Professor Atif, please unmute your phone or your computer. Yeah, so we can hear you. Professor Atif, can you hear me? If so, you need to unmute your computer.
Yes.
Cool.
Now, because this mute has to be from the host, because I'm trying always to mute my phone to hear me, but this can be arranged by the host.
All right. So sorry to interrupt
Okay.
now. So please carry on with your presentation. Thank you.
Yeah. Almost I finished my message, or I can also say it here. My message is for the whole audience that gene transfer strategy is a long way. It takes so much time and costs so much money also. So there is an alternative strategy that we can think about, and I can encourage the researcher to use it. Like, for example, it is known that inducible genes promote generating known molecules that can be generated under stress conditions, whatever which kind of stress, but it is a general molecules known that can helping plants adapt with the stress conditions. Like I have shown here in my presentation, like ascorbic acid, proline, glycine, betaine, in other papers also like abscisic acid, and so on. So why we are not starting from the end product of the genes, the end transcripts, which coming from the inducible gene under stress conditions. So we can use such molecules, especially we can find it as a chemical, artificial chemical, and can use it for foliar application under stress conditions, so we can help plants to adapt with that stress condition. So this was my message shortly. However, of course, I encourage, of course, to use the gene transfer strategy if the cost is no problem with it, and it is also helpful for the single genes and especially in biotic stress, like pathogen, kind of pathogen, and so on. But for abiotic stress, because under abiotic stress conditions, not single genes working alone, there is a huge number of genes, net of genes working together to promote generating a defined molecule to help the plants adapt with such conditions. So this was my message, and thank you for your attention.
Thank you, Professor Atif. So does anyone have questions, or would you like to ask during the discussion time? I think Professor Weedy is ready. Are you ready, Weedy?
Okay.
All right. Thank you, Professor Atif. That was very insightful. And I can see that with the strategic and further and more effective. So thank you for sharing that with us.
Thank you.
I will move on. Yes, I will move on to our third speaker, which is Associate Professor Weedy Sunayo. He is from Indonesia. A little bit of his background for you. He finished his bachelor degree in agronomy, his master degree in biotechnology, and his PhD. He graduated from the Göttingen University from Germany. He is now a lecturer and also the head of International Office and Excellent Program at the University of Mulawaman, Indonesia. He is working in the Faculty of Agriculture, and today his presentation will be the expression study of gene in Arabidopsis, and he specifically will talk about NOx genes, which I think we'll better leave to him. All right, Weedy, if you are ready, you can start the presentation. Thank you.
Okay. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone, everybody who join our tripartite seminar today. This is a very good time for us to share our scientific knowledge, because I know that Dr. Banyat and Dr. Atif is our friend during our PhD program in Göttingen University, and we talk together. Sometimes we eat together also during the lunch time. And now we hope that we can visit each other in Thailand and also in Tanta University to make our collaborations closer, and we can implement all our plan and also programs in the future and the next time. I would like to thank also for Vice Dean Professor Asad Derbala to join us to give remark, also to Dr. Tul from Prince of Songkla University, also my Vice Rector for Planning and Corporation, Dr. Buhari Yusuf. I hope that this not only one-time program, but we can continue with other useful and benefit program. I will start my presentations. I will start to share. So today I would like to talk about the expression study of genes in Arabidopsis, and I will take NOx genes, and this one is the part of my PhD project. And I want to emphasize to the protocol or to the procedure how to analyze and how to do functional study of genes, because we know that the functional study of a gene is still very interesting. Yeah, still very interesting. Many genetic and physiological process associated with environmental defense mechanism or many other developmental process is still unrevealed. So many people still work in functional study. So that's why in this time I would like to share my experience how to study and to do a serial protocol to analyze and the function of the gene. But of course, our presentation not completely until the protein function, or also in ex vitro examination. I will do and present limited until the transcriptomic analysis. And before we continue our presentation, I just to remind about the how a gene works, because this is very important. We want to study about the genes, but we have to know how a gene works. As Dr. Atif present previously, that's in eukaryote model, a gene contain promoter regions. Also, very important is intron segment and also exon segment. This is very different with prokaryotic model. That's the prokaryote model doesn't have an intron. So the process of information transfer is also different. Yeah, the gene will transfer the information through transcription to create mRNA, and then the mRNA will be a template for a protein for the protein synthesis, and then the protein can be a structural protein or can be a functional protein. The functional protein can be a hormone and also can be enzyme. The enzyme control all metabolism process in the organism. That's why a gene, we can say that a gene control the life of organism. And a gene can control a character of an organism. And forward the functional study, yeah, by functional study of a gene, we will know how genes and the protein can include function in the intact organism, and also to study the functional the functional of a gene. Usually, we use a mutant organism because by study the mutant organism, we know that if a gene is interrupted or inserted or inactivated by mutations, the nucleotide sequence will be also changed. And finally, the mutant often hold, and also the mutant will show the different and unusual appearance, like for the example in the fruit flies, white eggs or curly wings, or in the plant downward pointing calyx, and so on. And just as to figure out how the mutant organism can help us to understand function of a gene, my professor said that if you want to know the function of a wheel in a bike, please release the wheel from the bike. And what happened with the bike? Except for this picture, yeah, this picture is very exceptional because they can ride the bike without forward wheels, yeah, and the bike cannot walk because the wheel is released. If you want to know the function of a gene, you can try to inactive or to make mutation and look at what happened with the plants. If there is a change, if there is a mutation, if there is a unusual performance, unusual phenotype, or something like that. And from the phenotype, we can know the function of the genes. And why we use Arabidopsis thaliana? Yeah, in this presentation, I use Arabidopsis thaliana. Arabidopsis thaliana is weeds, small weeds. Yeah, small weed, and with small pot of fruits, we can call as a calyx. Yeah. This is like a very simple plants. Why we can call as very simple plants? Because containing only 10 chromosomes. It's very simple plants. And fortunately, all the genome has been completely sequenced. So the sequence of the genome of this plant all is known. And we can publicly access in the internet, especially in the Arabidopsis.org. Dr. Atif also saw how to access the information about the gene and so on. Although the sequence already known, but gene not at all studied. And the other thing is that using these plants, many regulator gene that's very important has been studied using Arabidopsis. Why? Because Arabidopsis is very small, low number of chromosome, and also the life cycle is very short. Only 1.5 months we can harvest the seed. We can imagine if we use tree. Tree is like 20 years, they start to make a life cycle to give a fruit and so on. It's very time consuming. So this is the easy one to use the Arabidopsis. And the study of functional gene in Arabidopsis has significantly contribute the understanding many aspects of plant growth and development. And the other one, why NOC genes? NOC gene is a homeobacter, connoted like homeobacter genes. Brief one is NOX. It's our family of homeobacter genes identified in all monocot and dicot. And a subset of this gene regulate meristem function in all higher plants. It means that this gene control many aspects in plants. And most of them is a regulator genes. It means that to study this gene is very important. And this gene not only in Arabidopsis, in almost all of monocot and dicots. And why we call as a NOC gene? Because this gene containing conserved DNA binding homodomain motif. This is very specific for NOC genes. Then this one, this concept DNA binding homodomain motif is always found in the transcription factor. We know that transcription factor regulate the expression of a genes. It means this one is a regulator gene. And based on the phylogenetic study, there are 8 members of NOC gene in Arabidopsis. This is only in Arabidopsis, divided into two subclasses, NOX1 and NOX2. The NOX1 comprise STM, NOT1 or BP, Brevipedicellus, NOT2 and NOT6. And the second class consists of NOT7, NOT5, NOT3, and NOT4, based on the amino acid sequences. And the most familiar and popular mutant of this gene is STM and NOT1. In the NOT1 mutant, the helix is downward pointing. It's very different with the wild type. With the wild type is upward, but the mutant is downward. And also the STM mutant is the failure to differentiation process in the meristem. That's why the shoot is inhibited. and result in the abnormal and sterile inflorescence. This is the phenotype of these two mutant. And for the expression study, I want to make a summarize, but it's very complicated and long. Yeah. So before we start for the expression study, first we have to visit the Arabidopsis.org. This one for Arabidopsis research. Yeah. And from there we can search everything about the information of the gene and the mutant and so on, because all of the sequence already there, already sequences, and this is publicly accessed. Yeah, we don't need like license and so on to access it. And then after we find our mutant or our gene, yeah, that we interested to, we can do identification and genotyping. There are some process to canamycin for identification and genotyping, canamycin selection, phenotype identification, tDNA and international fragment genotyping, and then we can do expression study using a real-time PCR. Before doing real-time PCR, we have to do primer design, RNA extraction, quantity and quality check of the cDNA, and then we do real-time PCR. And then from this real-time PCR, we can look at the expression of a gene. For the example, is in the tissue-specific expression, also in the targeted tissue, or based on the treatment. And from this one, we can study the function of a gene. This is, I just want to show, this is the website of Arabidopsis.org. We can input here, for the example, I want to look at the STM. STM with small letter, it means it's the mutant. If we use a big letter or a capital letter, it means it's a wild type, not a mutant. Yeah, this is a mutant. And then we will get information, everything there. Yeah, for the example, the number one, we can click here. This is the STM or shoot meristemless. Shoot meristemless, it means this plant file to form the shoot. Yeah, and then we can click here, and all information about the genes, about the mutant is there. The information about the gene, the insertion, the mutation, the phenotype, and also the sequence, everything there, and we can copy and paste. And we also can order to buy the mutant. Yeah. And from our experiment, we try to get all mutant of NOC genes from the STM, not 1, not 2, not 3, not 4, not 5, not 6, not 7. Also, we have to have the wild type, because as a control. Yeah, and we have to choose the ecotype is in the same background, because different background, different form, different shape, and different performance of the plants. For the example here, we can look at that this STM-GK is the name of the mutant of in the STM genes. How to do the mutation? They insert the tDNA in the first intron. For KNOC2, they insert tDNA, like Atif also mentioned about the tDNA, in the third intron, and so on. And we can look at the map of these genes in in the website. For the example like this, just to show you, this is for the STMGK1 and KNAT1. There are 3 intron here. Intron is the DNA segment that will not be transcribed to mRNA, will be spliced and throw away. Yeah, and then we can make the tDNA is inserted in this intron. Yeah, inserted in this intron, and for the KNAT1 is inserted in this intron. This is how to make a mutation in these genes. Yeah, this is the stem gene, and this is KNAT1 gene. And in the tDNA fragment, there is kanamycin resistant gene. They put kanamycin resistant gene for selection process. Yeah, for selection process. If they are a mutant, it means they will be resistant to the kanamycin antibiotic, something like that. This is the process how to select the mutant using kanamycin antibiotic. We do like a tissue culture. We put sterilize the seed of the Arabidopsis mutant to put in the medium. This is a Morisek and Skook media, and containing, of course, a kanamycin antibiotic, because the mutation using tDNA containing kanamycin resistant genes. And the result is like this. Yeah, if the mutant is sensitive, they look pale and white. If the mutant is resistant, it means that they will grow very well. And this one is the heterozygous one. It means that in the one pool of the seed, there are the sensitive, also the resistant ones. Yeah, and this one is the homozygous dominant, and this one is the homozygous recessive. And from this experiment, we can try to count how many seeds that is resistant. This is very important step to look at which one the heterozygous and which one the homozygous. Like KNOT4, KNOT4, KNOT5, this is already homozygous. Yeah, it means that it's already homozygous. We don't need to continue the next step for the identification. Yeah, why? Because for the mutant, if we want to do a functional study, we have to get the homozygous mutant. It means in the both allele, there is insertion. If only one, it will produce the heterozygous. The heterozygous one will not perform the mutation, the mutated plant. We have to get the homozygous. That's why we have to do selection to get the homozygous ones. And how to do? For the heterozygous one, we have to choose from the resistant seeds, the resistant plant. We have to do genotyping. I think Dr. Atip also saw this, how to process the genotyping. We can do PCR, using the specific primer for genotyping. And we can use this tDNA signal genotyping, and we just input the name of the gene here, the mutant here, the shock lines, the name of the shock lines. For the example, in this one, we already have the name of the shock line here. We just input this kind of number, and they will give us a recommendation of the primer for the LP, RP, and also LBP here. The LBP is to select the homozygous one. And from the PCR, we can run in a electrophoresis gel. And we can see here that for the example is the knot 2, yeah, and number 3, yeah, and the number 3 is wild type because only amplified in the LP and RP primer. And number 4 is exactly this is the homozygous one. The sample of or the seed of or the plant of the number 4 is the homozygous one. Because it's not amplified in LPRP, but amplified in LBA1, LP1. And the other one, if amplified in the LPRP and also in B1, it means this heterozygous. And the second knot 3, everything is in the LBA1, LP1. It means the all seed here is homozygous. This is for example. Yeah, this is a genotyping. So it means that we already get the homozygous seed. Yeah, it's very important to get the homozygous. And this is the for the knot 1, especially for the BP9, because the mutation is derived using the SPM transposon, not tDNA. So we can use the phenotype selection. If we plant and look at the inflorescence like this is downward pointing silic, it means this one is already homozygous of the knot 1, homozygous for the knot 1 mutant. Yeah, and for the STM also we can look at the performance of the plant is the inhibited shoot. It means that this one also the homozygous mutant of the STM. And from the genotyping and identification, we can look at here and make a summarize that after we buy the seed from the Arabidopsis.org, we can get the homozygous plants and we can start the expression study. So before the expression study, we have to do something that I think very long time. Yeah, during my PhD, I think I take around 1.5 years to do only the genotyping. Yeah, it's very hard to do genotyping. And then for the experiment study, we have to do real-time PCR. First step is RNA extraction and cDNA generation because real-time PCR cannot amplify the RNA. We have to convert the RNA to the cDNA using a retrovirus transferase, yeah, or we call it reverse transcription process. And then we can use the sample for the tissue specific experiment using, for the example, leaf, inflorescence, hypocotyl, and so on. And we can also use the sample for our targeted gene and tissue and also treatment, something like that. And after we extract the RNA, the most important thing is to check the RNA. If our RNA is in good quality or not. Like this, we can know that if the RNA good is, we can see the very distinct and thick bands in 20X RNA and also in 18S RNA. Like this is very, very good. And the mRNA will place in this because the size of mRNA is very variable. Yeah, there is a short, it is a long and middle and so on. So we'll place it. So we cannot see there because it's very will be smeared in here at the mRNA. So this is the result of the RNA extraction, and we run in the gel electrodes, and the result is very good. And afterward, we convert the RNA to cDNA. And after we get the cDNA, we have to make sure that our cDNA is good also. we call as a quality and quantity check. First, we have to make sure that the concentration for every sample is equal, because during the extraction, we will get the very different concentration. But we have to adjust the concentration to make sure is equal. Yeah, and then we can run in the gel electrophoresis, and we look at here the very distinct band here. It means that with the same size of actin gene, it means that the quality of the CDN is very good. And then we can continue with the real-time PCR. Using the real-time PCR, we have to design the primer first, because real-time PCR with the routine PCR is different. Using real-time PCR, we have to run very short fragment. Yeah, for the example, only maximum is 700 or 800 base pair. For the routine PCR, we can more than 1,000 and so on. Yeah, and then we can use for this sample is using this Roche Applied Science. We can use this website software to design the primer. But we have to pay attention about the template what we use. Yeah, for the example, I do this one. This is my problem during my study. That using this primer design, I got this one. That the primers in here. This is the upstream of the tDNA insertion here, and we have the result that the gene is not down-regulated. It means there's not mutation. And then after many time of trial, I changed the template of the primer design. I copy paste in the downstream of the tDNA, and we get the down-regulated, this one. The downstream, we get the down expression, because if we do mutation, it means that this gene will be down-regulated. If not, it means that our mutation doesn't work. Yeah, doesn't work. Yeah, and then this one, finally we get what we want. Yeah, so because in the transcription process, they still make mRNA from the beginning until this one, until the DNA insertion. So if we check here, the transcription still there, although will be truncated in here. So if we use this one, the expression still high. If we use this one, the expression will down and will very low, like this. Minus, it means this don't regulated, or the number of RNA very low compared to the wild type. And afterward, we do real-time PCR, the formula, the concentration, and the condition for real-time PCR. We can follow the company recommendation for the pre-denaturation and so on. And then we will have a result like this. For the example, before we do the real experiment for our targeted genes, we have to do the dilution experiment. What is the use of the dilution experiment? Because every genes have different performance for the real-time PCR. Sometimes they very high, sometimes very low. So that's why after the dilution process, it means, what is the meaning of the dilution? We make a dilution 1 time, 10 times, 100 times, and 1,000 times, and 10,000 times. Using this dilution, it means the concentration of the cDNA will reduce gradually. Yeah, and the graph is like this. And from the graph, we can look add the formula of the linear line formula. And for the example, this is the linear line formula. We get the efficiency here, yeah, and the slope y-intercept. And using this value, we can calculate the absolute concentration of the cDNA to convert the CP value, because real-time PCR only show the CP value. CP value is crossing point of this graph, yeah, the crossing point of this graph. For the example in here, yeah, in this is the number of cycles, something that, but they cannot show the real or the absolute concentration of the cDNA. So we have to convert to the absolute concentration using this formula. This formula is designed by the company. For my case is from the Roche light cycler 480. And then don't forget, or we have to pay attention about that. During the experiment, we have to have 3 technical replication for the real-time PCR and 3 biological replication. And we have also to do internal plate control, because between plate to plate, sometimes there are different conditions. This is to make a control. And I have said before that the real-time PCR, we can follow the company recommendation. And afterward, after the real-time finish, we can export the CP value to the Excel as our data. And then we can calculate the absolute concentration using this formula. And afterward, we can do the relative expression analysis by we normalize the expression to the actin-2 expression as housekeeping genes, and then we can make a absolute relative expression also to the internal plate control, and then we can compare each other. For the example, yes, this is my experiment that we can compare the expression of the STM gene between the mutant and the wild mutant. The expression of the gene is greatly down-regulate. It means that the mutation is very effective. And also the KNOT1 minus 244 times. It means that the mutation make the expression of NOT1 is very, very low. Also the KNOT2 here and KNOT3 here. But we look at here and KNOT4 and KNOT5. It's not down-regulated. It means that the insertion of the DNA does not influence the expression of these genes. So in the next analysis, I didn't include KNOT4 and KNOT5. Why? Because the mutation doesn't work. Yeah, because you know this one is for KNOT6 and KNOT7 also greatly down-regulated. This is very good for the expression analysis. And then for the example, we try to do specific tissue-specific expression in. I choose for the example STM, NOT1 and NOT7 genes in wild type plants. This experiment want to know in which part of the plants this gene is expressed. And we know here the blue one is STM, the red one is NOT1, NOT7. The blue and the red one, it means STM and NOT1, always express in almost all part of the Arabidopsis plant except the leaf. We assume that if the part of the plant containing like wood, yeah, like wood, or silent cells, the expression will be high, yeah, because this gene is specific expressed in the meristem cells in cambium, something like that, yeah, and cambium also in the shoot. In the leaf, no expression is look here. And then we can do another expression analysis for the example to look at what is how the expression of all 7 gene, all NOx gene in the 4 mutants. I did also the double mutant here, STM and KNAT1. Both of these gene also don't regulate it using mutation. I do the crossing between the one mutant and other mutant. And then we look at here in the STM-GK or in the STM mutant, the STM gene don't regulate it. But the other gene is normal. And the NOT1 mutant, only the NOT1 is downregulated, but the other gene is normal. And in the double mutant, both of the gene is downregulated. It means that our double mutant is really the double mutant. And the other gene is little bit increase. And for the KNOT7, the KNOT7 gene is downregulated, but the other gene is increased, upregulated. This is very good question to do next analysis. What is the function of this KNOT7 and how it can influence the other gene, the other KNOT genes. And then I think this is just to show you how to look at the function of STM and KNAT1 to the other gene. For the example, from the website, from the internet, we estimate that this 2 gene is a regulator for the cellulose biosynthesis genes. And then we try to look at the cellulose biosynthesis gene. For the example is Pmax NAM1, LTP4, FLA11, IRX1, IRX3, IRX5, and so on. And we can see here that this is in the wild type, and this one in the STM mutant and the KNAT1 mutant and the double mutant. And we look at this, the expression of this cellulose biosynthesis genes is decreased and if very greatly downregulated in the double mutant. This is my presentation. I do hope that my presentation can give benefit for a researcher that want to do like a functional study in gene analysis. Thank you very much.
Thank you
for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Weedy, for your presentation. And I think now we are moving on to the discussion session. Regarding the discussion session, if any of you have the question, you can, if you would like to, you can ask the speaker directly now. And then just now during your presentation, we have a question from audience for Professor Weedy. Right. I think the technician staff, they are preparing the question for you to share with you. Okay, let me. The question was from Miss Anasta Faradilla from the Agricultural Department. And then her question, I'm gonna send it to your chat, Weedy. Then you can read and sort of like share with us.
Okay, I can answer directly.
Yes, please.
Okay, the question is, in plants, we can make the gene become inactive. So how to make sure that the gene become inactive? Yes, I think our explanation, most of our explanation talk about the mutants. We can make the gene inactive by a mutation. We make insertion of the gene. And in our case is in the Arabidopsis we use a tDNA insertion. If the gene is inserted by DNA fragment or another fragment, the form of protein or maybe the transcription will be interrupted, and the mRNA will truncated, not fully mRNA. It means that the expression of the gene uh, will be inhibited. This is the how to make the gene is inactive. How to make sure? To make sure is using if I said that using expression analysis. If the expression of this gene in the mutant is down-regulated, it means this exactly the gene, the inactivation of the gene works. Something like this, my answer.
Uh, Miss Anastasia, are you happy with the answer, or you would like to ask more?
Hello, everybody. If you can hear me, please. I think there is a technical problem again. I cannot hear you.
Oh, we can hear you.
You can hear me, oh?
Yes, we can hear you.
Yeah, you can hear me, but I can't. I cannot hear you.
Right.
Yeah, I'm speaking and I'm not able to listen to you.
We can hear you
Okay.
perfectly, Professor Atif, yes. Professor Weedy, I think we have more questions for you in the chat. Can you see?
So directly answer, Achan?
Yeah, please. Because everyone can see the question in the chat now.
Yeah. If we want to see the difference in plants, we can make the gene become inactive. So how to make sure that, I think it's already answered. And what is the striking difference of the phenotype between wild type and the mutant? The second, how the response of knock gene to the biotic stress. Okay, thank you very much. The first question is, we can look at for the example is STM gene and also KNOT1. It's very significant differences between the mutant and the. I just already showed the picture. For the example, the KNOT1 is the downward pointing helix. This one because of the mutation of the NOT1 gene. For the STM is the inhibited or undifferentiated of the meristem. The shoot is inhibited. They cannot grow normally. This one is because of this very typical for the STM mutation. And how the response of NOT gene to biotic stress? According to my knowledge, that NOT gene control the development. Most of them is control the developmental process. For the example, how to switch in the cambial process, how to switch and to do differentiation after the initial cambial cell, how to go will go to the phloem or go to the xylem. The control of this process will be conducted by the NOC gene, especially STM also not one genes. The other process is how to switch the gene to make a shoot and also to make a flower, for example. This is the leafy gene or something like that. Many knock gene control the switching the developmental process, but not the abiotic process. Thank you very much. Maybe the study, the role of the knock gene to the abiotic stress defense mechanism is still no until now is very limited and very little. This is my answer.
Yes. I really want to give you a break, but then you have the third question in the chat. The question asked, is the knock gene its responsibility for what happened in the Arabidopsis? Can you see the question in the chat? Please unmute, Professor Weedy. Professor Weedy, please, we cannot hear you. Can you unmute?
Okay, sorry. Okay, so knock gene also work in other plant, not only in Arabidopsis. But why we do the study in Arabidopsis? As I mentioned before, that's the plant. If we do experimental design and functional study in the tree plant, for example, the life cycle is very long time. We need to make sure the homozygous seed, something like that, to produce it is very long time. Yeah, and then using Arabidopsis, we can make homologous comparisons, almost the same but different, something like that. And we can make homologous expression. And afterward, many knock gene in other plants, yeah, is found also. Yeah, arbor knock and so many knock gene in other plant also. And it means that it can be applied for other plants. But for the beginning or initial study, initial function study is in the Arabidopsis. Why? Because the information of the sequence is already there, already completely studied in arabidopsis.org. This is my answer.
Yes, thank you very much. Actually, I have a similar question for Professor Banyat. Can you hear us?
Yeah.
Yeah, because you are studying like fungus, like fungi for the production of ethanol in sort of like the palm oil tree. For those people probably in the Indonesia or in Egypt that they have different plants, can they apply your technique to produce like a value added from the agricultural waste?
Yeah. Thank you for this question. Actually, the wood chemical contained at the same in the world. That three main: cellulose, hemicellulose, and lignin. Cellulose and hemicellulose consist from sugar, and the big amount come from cellulose. Cellulose chain is consists from glucose, one molecules to another molecules. The contents about 50% like that. If you can digest it, that we call mega hydrolysis. You take the sugar and make the fermentable sugar to be bioethanol. If Ajay work with the mango wood tree also can convert to bioethanol. From Weedy, I think we not much different. We are in the rainforest.
Yeah, Thailand and Indonesia.
when there sometimes I saw many palm oil plantation, you also can use this. Even the sap from the stem, like a trunk, like we call a big cassava. No, big um, something sweet. What is that? Sugar to produce some sugar.
The sugar cane?
Yeah. Sugar cane that we can say a very giant sugar cane. One piece of this and then long like this, you can get about 1 liter or more than 1 liter, and then I try to produce bioethanol, and it reached the minimum requirement like from sugarcane bioethanol. This also possible, and in southern part of Thailand, I think has a very high potential to convert this plant to bioethanol.
And I think it's a good idea as well. Like instead of throwing it away, we can
Yeah.
something and sell for some money. But then we have to think about the cost. I would have a question for Professor Atif. Like your work is about gene transfer for to help the plant against weather changes. Do you have some example like we can follow? Like what sort of character in plant that you so like modify to improve the weather changes?
Okay, I would like my message in my presentation was to say that climatic change are correlated with many stresses because if the temperature is high, the sunlight is very also high. This can be induced other stresses like osmotic stresses, drought stress, salt stress, and this all together can induce huge number of genes. So abiotic stresses all related to each other. So in abiotic stresses, we can follow single genes that can contribute with other genes in helping plants to adapt to stress conditions. Yes, during my PhD studies in Germany, I was interested in salt-inducible genes. So I worked at single genes. And yes, it was promising that these single genes somehow helpful for plant adaptation against salinity. But as Dr. Weedy, for example, says in his presentation that he takes more than 1 year just for genotyping the single genes. So the work in that field is very hard. So, and the steps coming from the beginning that if you have seen some genes were expressed under defined stress, you have to be sure before going further with the study that this gene in other organisms are stressed as well. So the message is under abiotic stresses, huge number of genes are working together for helping plant adaptation.
I think we have a signal loss, but I'm sure Professor Atif will come back very soon. Ajahn Banjat, just now someone asked you a question.
Yeah.
Okay.
Yeah. She's coming.
Okay. So the message is if we start from the end product of inducible genes, it's special molecules like I have shown in my presentation, like ascorbic acid, proline, glycine, betaine, abscisic acid, alum sulfuric acid. So many molecules can be generated under stress, and these molecules can act as artificial chemicals. So we can start from the end using these artificial chemicals, artificial molecules as foliar application to help the plants adapt with this. At the level of single genes, of course, it will be poor if you have the budget, if you have the facility in your lab to take for proof that special genes are really helping plant adaptation in defined conditions. I hope that my message is already really. I hope.
Right.
Yeah, have you got my message now? My answer is clear, or?
I think we have a little bit of problem with the signal, but I think it has been a really good discussion that we have after the presentation. But due to the time that we are running a little bit behind the schedule, I think we might wrap up our webinar today around this point. For the audience, for the attendance, we have posted the attendance link for the registration in the chat box. So you can click on the link and then put your information, and the system will generate a certificate of participation for you all, like automatically. Is it okay for the speaker if I would like to wrap up the session about now? I can see Weedy like nodding his head, but I can't hear him. Is that okay, Weedy? Okay, right. First of all, I would like to show my appreciation for all of the speakers that you have been sharing with us. Like your research, you spent like 3 years, 4 years to find out this thing, and then you just share it with us for free. So, and then like, I can see you have a little trick on the research, the technique, and then your insight information of the vision that you have on the research field. I think we all appreciate that. On behalf of all the audience, I would like to thank you all the speakers for their time. I also would like to thank you our universities that allow us to have met today. The secretary that has been working really hard, the IT technician that have been working like, you know, behind the house, like every minute they're just working, and then they are on their feet all the time. So I would like to thank you them for that. And nevertheless, like our webinar will not be as good as this time if our participants like don't choose to spend time with us. So thank you for your attention, and then thank you for choosing to spending time with us. All right. I think on behalf of everyone, I would like to say thank you, and hopefully we will see again in the next webinar. Okay?
Okay.
Thank you.
Thank you very much.
Thank you.
Very nice, Achan. Anya, thank you very much.
Thank
Nice
you.
to meet
Thank you.
Thank you, Atif.
Atif.
Professor,
Thank you, Bania. Thank you.
some other time.
Oh,
Yeah,
boleh
but
visit us in Mulawaraman ya after the COVID-19
Yeah.
finish.
Yeah.
Yeah. And I promise to finish the MOU as soon as possible.
Okay. With Aceh, huh?
Our selector already here.
And
Yeah.
Baniat, we need
Mhm.
also a collaboration protocol. Yeah,
Uh-huh.
I started with Weedy already to handle such protocol. But with you, yeah, I need a formula or something like that. You can send it to me. Yeah?
Yeah, okay. You can write me via MSN or whatever, huh? And then I will manage for you.
Okay.
Okay.
Yeah,
Feel
but
free to leave. I think people are afraid to
one thing. Yesterday, I just only make kidding. Really, if you still remember that I put Aceh University logo behind, but now it's come true.
Exactly in the center, yeah. Exactly in the center. Because your asked me that the ADIP logo have to be in the center.
Professor Weedy, is the certificate generated automatically now or when?
Yeah, when they fill the form and then the certificate directly to send to their email.
Okay, so it's sort of like
So the people have to fill. All of participants have to fill the presence form, and then they can leave the meeting. Afterwards, directly the certificate. For you or we will make a different certificate because for the
I can have
speaker
two because I register as well.
you register also. You can also register for the participation, but we will also for the speaker and also for the moderator.
Yes.
Very good.
Yeah.
Okay, feel free to leave. About 248 people are afraid to leave.
Yeah, 264 people. It is, I think, very great number. Yeah.
Yes, yes. And very informative and then very interactive.
Yes. Thank you very much, Achan. Very nice for the moderator.
Thank you.
Thank you.
And thank you, Achan, Professor Asad.
Thank you.
Oh, Professor
Thanks very
Azad, we
much
hope
for
to
your join.
see you.
Hopefully, we can visit Egypt next time.
Okay.